So welcome everyone to the Botany Speakers Series of California Botanical Society. Today we're going to hear from Irene Liao on exploring monocot nectary evolution through a genetic characterization of banana and asparagus gynoecium at the single cell level. Irene received her PhD in biology from Duke University with Mark Roscher, where she studied the evolutionary genetics of reduced nectar production in a selfing morning glory. Irene is a California native who grew up in the San Gabriel Valley. Her love for plants strengthened during her first research experience at UC Berkeley, where she worked in Chelsea Speck's lab to develop functional genetic tools for onions. She also worked in two maize genetics labs, one at UC Berkeley and the other at Cold Spring Harbor. Most recently, Irene is an NSF postdoctoral fellow in biology, sponsored by Drs. Karen Sears at UCLA, Luke Nikolov at Indiana University, and Lena Heilman at University of Kansas. Irene is interested in floral trait evolution, and as we'll hear about today, her focus is on nectar and nectary diversity. She's also used quantitative and population genetics to understand variation in nectar between species, and she uses single-cell transcriptomics to identify candidate genes underlying monocot nectary development. Today's talk will be interesting and maybe a little different from our usual talks, as we'll be hearing about nectaries in banana and asparagus. But we'll learn about current advances in single-cell transcriptomics, and hopefully we'll hear about how these results can give insight into nectar evolution in other plants, including California native plants, perhaps. And Irene is on the job market currently for positions in California, so reach out to her if you're interested in her work. Okay, take it away, Irene.
Thank you so much for that lovely introduction, Adriana. And yeah, thank you for being here and for having me. So what I would like to do first, let's see how to advance. Here we go. And in case I need a laser pointer. Okay, here we go. So this is a general outline of what the talk will be like. I want to give a very broad introduction into nectar and nectary biology. I think most of us may not need this, but I just find it so fascinating. I think it'll be a great way just to be thinking about nectar and nectaries. And then touch base a little bit on the work I did as a PhD student, looking at the mechanisms underlying nectar differences between two sister species. And then later really just focusing on the candidate genes underlying monocot nectary development. So the structures that are then producing nectar. And this is a talk that I had given over the summer, so hopefully that's not, that it works for this audience as well. So hold on, I actually have something that I need to, oh wait, okay. All right. So I don't need to belabor this point, but I like to start off here talking about how nectar is really at the center of so many important plant-animal interactions. Nectar itself being the major reward and also food source for many of these animals, as you can see here. In part, that's due to, in exchange for services such as exchanging pollen for pollination, or even defending the plant, as you see here with this ant and these extra floral nectaries. And so it really, these interactions really benefit both organisms, both partners. Nectar itself is an environment for microbial interactions. There's a really interesting microbial community that's happening in there as well. And I put this picture of a beehive and honeybees because just to remind us all that honey is a concentrated form of nectar, but also some of what these insects are going when they go visit nectar, it's not just food source, but it might actually be benefiting their gut microbiome. And that's actually an area of interest. Most recently, there was this review talking about some of the ways that the nectar microbiome can actually affect pollinator health. So I just mentioned the gut microbiome of these insects, But they can also, the microbiome can also play a few other roles, such as increasing the foraging behavior in some of these plants, or even increasing the attractive signals that the plants are emitting to attract all these other pollinators and so forth. So if you're really interested in this, I would definitely check out this review. The other thing to note is that nectar itself, it's not just sugar and water. So those are the major components, but there's also other metabolites that are found in there. So amino acids, all their volatiles, and even compounds such as this right here that was just described a few years ago called nicotine, which confers this red color in this particular plant called nicotine from Mauritius. And you may be asking, well, why even have colored nectar to begin off with? And it is proposed that it is a cue to attract pollinators. So on Mauritius, there's other plants that also have colored nectar, and it is a cue for some of these lizard pollinators. So that nectar itself, it's quite complex. There's all this really interesting chemistry, but the structures, the nectaries themselves are also super interesting. And that is, you can find them on all different parts of the plant. So you can find them here on the sepals, these tepals. Here, it's a nubbin at the base of the flower. You can see these elaborate structures here, some even these tubular structures in Cydia, and some that just look like this ring that's found around the ovary. So there's quite a lot of diversity, just the placement and even the morphology, the size of these nectaries. But even when you look at it from an anatomical level, you can see that there's diversity there. You have trichome nectaries. You also have epidermal nectaries here, where the nectar seems to just burst out of the cells. And as you probably saw this earlier, and even in this case, you have 3D structures, where here, these little circles are basically, of course, those are nectary stomata. They're open, and that's how the nectar is then secreted out to the outer surface. And so this is just a overview of what I just mentioned. There's just this vast diversity of nectaries. And I'm only just touching on floral nectaries here, right? So again, just saying that they're found on all different parts of the plant. I mean, all different parts of the flower. You see what's colored in orange, those you see the different sizes, the shapes, and that's what really fascinates me, seeing all of this diversity across all of these different flowering plants. And how did these even all arise? So that was just a quick overview of nectar and nectary biology and the diversity and the range of questions that are being asked currently and still being asked. But I want to get into a little bit of my PhD work just very briefly, because this is how I started being interested in nectar. And kind of strangely, maybe not so strangely enough, I was actually interested in plants that don't produce a lot of nectar. And so there are actually, these different cases where these plants don't produce very much floral nectar or no nectar at all. And that is usually due to these transitions to abiotic pollination, such as wind or water pollination. There's a shift in primary pollinator rewards. So it's now oil, scent, or pollen that these pollinators are looking for as their reward and not nectar. And then there's also this other case where there's the shift in mating system. So going from outcrossing to being highly selfing. And usually what happens is in that transition is you have a lot of other morphological changes that occur. So the outcrossing species tend to be a lot larger than that of the selfing species. Selfing species isn't just only reduced in overall flower size, there's also reduction in pollen, scent, nectar, and at times, depending on the species you're looking at, also a reduction in color. So the species pairs that I did the study looking at this transition in mating system were these two morning glory species, Icobia cordata triloba and Icomia lacanosa. And these happen to be crop relatives of the sweet potato, but they are also quite weedy, and so just keep that in mind. And so the major question here I was asking was, how did reduced nectar production evolve in a highly selfing species? And you can probably guess, just looking at the morphology, Chordata triloba here is the outcrossing species, and Lacanosa here is the very highly selfing species. And so I had measured nectar volume, nectar sugar concentration, and saw that, yes, in fact, both of these two traits are reduced in the highly selfing species. And so with other colleagues in the lab, what we did was we did a lot of quantitative genetic approaches to try to understand some of these mechanisms. So one of the questions being, well, is it natural selection or is it genetic drift that could be leading to the divergence in these traits in the two species, and not really going into how this QST-FST test works. Generally, the pattern is if you see your QST, which is the quantitative trait genetic differentiation, is greater than that of FST, which is the neutral genetic differentiation, this is a pattern that suggests that selection probably played a role. And that's what we see here for both nectar volume and nectar sugar concentration. So I also did more quantitative genetics, basically using a mapping population of crosses that were made between these two morning glory species, and measured a few different traits, including some nectar traits, some seed traits, and quite a few different flower traits. And basically trying to ask, well, how genetically correlated are all of these traits? And what you see here is that you have very high genetic correlations amongst the seed traits, high genetic correlation amongst the nectar traits, and a high genetic correlation amongst the flower size traits. And each of these genetic correlations are the average correlations are much higher within each of these modules than any of the genetic correlations between any of these traits. So this is suggestive that the evolution of these nectary traits are likely independent of these floral size traits. Let me see. And then finally, I was really interested in trying to see, can we identify some of these genes that are important for the differences in nectar production, so nectar volume, nectar sugar concentration. And oftentimes when you try to do these sorts of studies, there's just a ton of genes that you're sifting through. But what I did was I combined results from that first study using quantitative genetics, used a bit of transcriptomics, and also combined it with population genomics, all of these to filter down to about 200 or so genes that likely explain these differences in nectar volume and nectar sugar concentration. So that is still work in progress, but hopefully I'll get that manuscript submitted soon, so you can see what some of those genes might be. So that's what I did for my PhD work. But that was all really looking at the nectar itself. And what I was starting to become more interested in was, well, the nectary, the structure that produces the nectar. And so then going back to, before I go into all of that, I want to talk about the key players, the people who I collaborated with for this work, where then I started looking at nectary development in monocot species specifically. And so I was, as Adriana had mentioned, I was co-sponsored by three fabulous mentors: Luke Nikolov, who started at UCLA and then moved to Indiana University, Karen Sears at UCLA, and Lena Heilman at the University of Kansas. And so this was all done as part of an NSF postdoctoral fellowship. And actually, this was also done under the collections track. And so I was able to make use of collections from the Huntington. So I really wanted to highlight Sean Lamar, who was instrumental in providing or gifting material for me to work on, and Michaela Roos, emeritus professor at UC Riverside. So without them and without them gifting me these material, I would have nothing to show you all today. So let's go back to what I had ended up with in talking about nectary biology, and that is reminding you that there's this great diversity across angiosperms. And really, that is the motivating question I have is really, well, how do these diverse nectaries form? And to really tease apart this question, I'm taking, perspectives from evolution and development. So really thinking about, the evolutionary angle, comparative approach, trying to understand the diversity of all these structures. But then at the development side of things, to see, well, how does the nectary form from when it starts, all the processes that happen while it develops, and then at the stage of maturation where then this nectary is secreting nectar. And so the link that I'm making between evolution and development is by taking a genetics approach. And so I'm reframing this question to be, well, what genes contribute to nectary development? So we do know a little bit about this already, mainly coming from the core U dicots, specifically in Arabidopsis and Brassicaceae species, where you have nectaries that are found at the base of the flowers as these little nubbins. And so here, this transcription factor, Cas was found to be really important in initiating nectary development. And this particular transcription factor has also been found to be highly expressed across other core eudicot nectaries, such as cotton or even petunia or tobacco, where you see they have a different type of nectary here, where it's kind of more like a ring that surrounds the ovary. But when you step a little bit out to look at the non-core eudicots, such as Acalysia or columbines, where they have their nectaries at the tip of these petal spurs, it's actually another transcription factor known as stylish that is important for initiating nectar development. So we know that we have some clues, we have some transcription factors that are important, but we don't know about this in some of these other lineages. And the one that I'll be focusing on today are monocots. And so before I even go into looking at, what sorts of genes could be expressed, I need to show you, well, what kind of nectaries they have. Many nectar-producing monocots have what's known as these septal nectaries. So this is an example from Agapanthus. And if you tear off the tepals, this is the gynoecium. And if you squint a little bit, you might be able to see a droplet of nectar right here, but you can't see the nectary actually. The nectary, in order to see it, you need to take a transverse section and then see where the nectaries are found. And so the nectaries are found at the margins for all of these carpels, actually fused, or it's the unfused regions. And so the nectaries are on those margins, and the nectar gets secreted into these bases, depicted here in light purple, and then it makes its way to the outer surface through ducts or slits. But not all nectar-producing monocots have septal nectaries. There's some exceptions, including in the Lineages, where you have the nectary found at the tepal. And in Zingiberales specifically, you have the nectary that's now protruding out of the gynoecium, and it's found on top. And it's a 3D structure there. So these are what's known as epigynous nectaries. So if you map all of these traits onto a phylogeny like this at the order level, what you see is that in almost all of these orders in monocots, there's at least one species in them that still retain the septal nectary structure. And then there's other lineages, specifically Lineages, where now you don't have septal nectaries and have tepal nectaries. And there are multiple instances of loss, most notably in the grasses, in the Poales, and also palms, but also some in the Lismatales, and those are shifts to either wind pollination or water pollination. And so by seeing all of this mapped out, it makes it a really fascinating system or group to really try to understand, well, how has these septal nectaries been conserved across these different lineages? What is the case that led to then the transference of the nectary to the tepal? And then also trying to understand, well, how did these nectaries become lost in these other lineages? So just want to focus here on the septal nectary, and again, seeing this pattern where you have, almost all orders have at least one species that still have this structure, it is thought that the structure likely evolved only once in monocots from the split from Asparagus, and that this is a conserved structure. And so given that hypothesis, the question I have is, well, if it is, conserved, is it also conserved at the genetic level of the genes that are contributing to nectary development? And so to start off with, before I can even answer all of these questions, I need to just know, well, what genes actually underlie septal nectary development in monocots. And so here, what I'm going to do, I'm going to talk about the approach that I'm taking, and that is the single cell sequencing approach. So I'm going to try to talk about it first by using this analogy and of using this fruit tart, and then later talk more specifically about the steps for making single cell libraries and also the analyses associated with it. So you can imagine this fruit tart being your tissue of interest. So in my case, the nectary. And as you know, tissues are the building blocks of tissues are cells. So you can imagine each one of these different fruits are different cells. And the thing to note here is that these fruits are arranged in a specific pattern, and that's also how cells are usually arranged or patterned in tissues. And so typically when people are trying to understand what genes are being expressed in these tissues, what you do is you're going to take your tissue and you're just going to grind it all up. And then you just look at, well, what genes are being expressed. And so you can liken that to a smoothie where you're just like taking all these fruits from the fruit tart and just blending them all up together. But as you know in the smoothie, and also when you grind everything up, you lose that information of where those genes, which cells those genes are actually coming from, where they're being expressed. You are getting basically now an average expression of all of these genes altogether. So what single cell sequencing is doing is rather than having all of these genes all blended up, you don't know which cells they actually come from, it's actually now retaining that information. So that's what you see here. So when you do single cell sequencing, you get to know, when you're looking at your genes, you're trying to figure out, well, what cells they're coming from. You have that information now. It's all maintained together. And what you can do then computationally is sort them out based on similar gene expression profiles in each of these cells. And so that's what you see here. So one thing is that this is quite powerful. You now know where these genes are actually being expressed at the cellular level. But one caveat with doing single cell RNA sequencing like this is that you lose the spatial context. You don't know exactly what cells or where those cells are actually located. So you don't know that these, in this case, these strawberries are actually, in the outer layer, for instance. So just something to keep in mind as we talk about this. So now going back to plants, how do you actually do single cell sequencing on plants? Well, first you need to get single cells. And two ways to do that. One is to get protoplasts, which is basically plant cells without your cell wall. So you're using enzymes to digest those cell walls out. And this can be pretty precarious sometimes because you can see they're pretty fragile. They don't have those cell walls anymore. Another way to try to get single cells, or not really cells, but really actually the nuclei. In this case, I'm showing you a picture. These are DAPI stained nuclei. So you can do sequencing on either whole cells or nuclei. And in my case, I extracted nuclei for my study. And once you do that, now you need to make single cell libraries. So the most popular platform nowadays is this 10x genomics platform. And just to walk you through a little bit of how that works, what they have is a tube that has a bunch of different barcoded gel beads. And basically you take those beads, you put it through a microfluidic device. You also then have your cells that you're feeding through this microfluidic device to then generate these bubbles known as gems that are combining your cells and your barcoded bead together. And once you have formed these little gems, these bubbles, now they're, all your cells are isolated with one of those beads, and you can have all the library making processes happen in there. So like a reverse transcription, amplification, and so forth. And once you have that, because all your genes that are coming from that one particular cell is now barcoded, you can make the mix. Basically have a mixture of all these different genes, and you can just sequence them and then computationally be able to assign your genes to specific cells. And once you have that information, typically what people do is generate what's known as a single cell atlas. And so here, each dot represents a cell. And what you do is you cluster these cells together based on similar gene expression profiles. And that's what you see here in all of these different colors. One thing to also mention is that this is a multidimensional data set. And so this is all then compressed into 2D, what you see here. And then oftentimes after you have figured out, your cell clusters, people tend to try to find, well, what genes are highly expressed specifically in this cluster. So you're basically doing differential expression, gene expression analyses between cells. For instance, looking at all the cells in cluster 11, you're looking at those genes and you're asking which genes are highly expressed in cluster 11 versus all other genes from all of these other clusters. Okay. And once you find those that are really highly expressed, those are these top rank genes that you find. And the reason for doing that is these top rank genes could help you infer the identity of the cell cluster. So for instance, if there's something that's really highly expressed, you look it up, look up what the homologous gene is in Arabidopsis, that can lead you to infer maybe it could be, an epidermis, or it could be vasculature or so forth. So that's an overview of the approach I was taking. So let's get into the species. And so the first species I am introducing you to is banana. And so this is from the Huntington. And here you can see pistillate flowers that are open first in banana. And underneath all of these bracts, you will then get development of these staminate flowers shown here. And so if you take a longitudinal section, you'll see actually the nectaries found spatially differentiated, where you have the nectary found on the top part of the gynoecium, and at the bottom part, the base, you don't have any nectary over there. And if you take a transverse section, this is what that septal nectary looks like. So I'm taking advantage of the spatial difference and where the nectar is found to try to apply it to my study here, to try to find cells that are specific to just the nectary. So what I did was, from the staminate flower, I made a single cell library coming from just the top region, and I made a separate single cell library coming just from the bottom region. And the idea here is that the cells that are coming from the top region should include nectary cells, and the libraries from the bottom region shouldn't have very many or very little nectary cells. And so I can basically compare these two to try to find cells that are just coming from the top region, or cell clusters that are just coming from the top region. And those then should be my nectary cell clusters. And later on, what I can do is try to find the genes that are highly expressed in those nectary cell clusters, and those would then be some candidate genes to look further into. So I did this for the staminate flower, but I also added one more library that came from the top part of the pistillate flower, and that also included some nectary cells. So once I got all the data, I made single nuclei atlases, and that's what you see here. They're all clustered from their specific regions. So I was able to get about 5,000 or so nuclei from the base, the top parts of the staminate flowers, and about 800 or so from the pistillate flowers. So this looks good. You have different cell clusters, But what I'm really interested in is, well, once we combine all of these together, which cells and cell clusters are just specifically made up of cells coming from the top region of the gynoecia. And so to do that, I had to integrate all of this information or combine them all. And so here, anything you see in orange are cells that are coming from the top region, and anything in this lavender purple are cells that are coming from the base or the bottom region. And what you can see here is that there's some cells that are both orange and purple that overlap, but you also see clusters where you only have orange cells and very little of the purple cells. Another way to take a look at this is the clusters colored by similarities in their gene expression. And so that's what this cluster is here. And what you can do then is if you compare these two, I've circled the cell clusters that seem to be just specifically made up of cells coming from the top and have very little to no cells coming from the bottom. And these are then my putative nectary cell clusters. And this is also just another way to convince you to show you that, yes, in fact, these putative clusters, if you look here from the base, you have very little to no cells coming from the base, and it is actually quite enriched in cells that are coming just from the top region, from both the pistillate and the staminate flowers. So what are some of these genes? So I'm going to show you 3 of them, but first I want to walk you through how to read these. So here, any of these dots, these cells that, these genes that they have high expression in these cells, it's going to be in this dark blue purple color. And anything that's in this gray color means there's little to no expression of that particular gene. And so the 3 that I'm highlighting here are agamis, where you see high expression in these 3 cell clusters, YABI2, which seems to be very specifically highly expressed in this cell cluster, and then sucrasynthase, which is also quite highly expressed in these 3 cell clusters. And of these, sucrasynthase is really quite interesting because if you think about what is the function of a nectary, it is to produce sugary liquid, right? The nectar. And so it's quite promising to see some of these metabolic genes that are involved in sugar processing. I'm going to just show you this. This is quite overwhelming, but here's also just a few other genes that are also highly expressed, some specifically in these clusters, and some that were also common across these clusters over here. And again, I see some transporters, but also then the sucrose phosphate synthase genes, or for instance, the cell wall invertase gene. And these are genes oftentimes when you're looking at nectary development or looking at transcriptions from nectaries, you see these genes popping up. So again, making me feel like, this strategy to try to find cell clusters that or maybe nectary seem to be quite promising. And so as a summary, using this single cell sequencing approach, I was able to find some of these candidate cell clusters that could be associated with the nectary. Again, unfortunately, I don't have spatial information. That's something that is lacking. But even then, just computationally looking at what genes are very highly expressed, I am picking up on some of these genes associated with sugar processing, and also some transcription factors such as agamis and agamis like 11 and YABI2. So I want to then step a bit out and go back to the question I had started off with, and that is, well, do these monocots that have similar nectary structures, do they have similar gene expression profiles also in the septal nectary? Or in other words, do these septal nectaries have a common core set of genes that underlie that septal nectary development? And so the other species I'm looking at is asparagus. And I just want to remind you that asparagus and banana are quite divergent. It split around 100 or so million years ago. So obviously, when you're doing these sorts of comparisons, a lot of different things have happened genomically. There's a lot of gene duplications, loss, and so forth. So it will make comparing these two species genomically a bit tricky. The other tricky thing is trying to do this at the single cell level. Thankfully, people are starting to develop tools to try to do these types of comparisons. And one of the ways is this pipeline known as this orthologous marker gene groups, or OMG for short. And so what people do, what this pipeline is actually doing is that it's taking all of those highly expressed genes in all of the clusters from one species and then comparing it with all of the highly expressed genes from all of these different clusters from the second species, and basically counting how many of those genes are similar or homologous. And then after doing those counts, trying to figure out how many of which pairs, right, actually have the highest number, that is also statistically significant, and statistically significantly associated. And so that's what's represented here in this red outline box. And so here, once you find something like that, for instance, here for species 1, cluster 14, and cluster 9 in species 2, it seems like they might have a lot of genes that are shared, and it may suggest that these two cell clusters are homologous with each other. So first, I want to show you what these asparagus flowers look like. So if you let your asparagus continue growing, they might develop flowers, and these are the female flowers. And then this is what the gynoecium looks like, and you can see the nectar being secreted. And if you take a transverse section, this is what that looks like, and the arrows are pointing to the septal nectaries. And so these sections here were made by Selena Vanko, who is a former master's student at the California Botanic Garden. And here's a close-up of that septal nectary. So one thing to note with these asparagus flowers is that they are much smaller than those banana flowers that I showed you earlier. And so in this case, in generating these single nuclei and trying to isolate them, I actually took the whole gynoecium and across different developmental stages. And so from that, I was able to get about 4,000 or so nuclei and generate this atlas. But because, in this gynoecium, there's so many different cell types, you're also getting stuff from developing ovules, the style, the stigma. I don't know where all these cells are coming from, right? What clusters they're actually, what the identity of these cell clusters might be. So I'm going to be using this OMG approach I had mentioned earlier to try to find homologous cell clusters between the nectary, homologous nectary cell clusters in the asparagus by using information from banana. And so that is the question, and that's what I'm going to try to do. And I just want to show you an example first of this approach and how good it is at finding homologous cell clusters. And so here, this is an example of the epidermis cell cluster, where it was identified that banana cluster number 6 is homologous to asparagus clusters number 4 and 8. And that was due to the fact that they had these shared genes. And some of these, if you recognize, especially ATML1, it is a gene that confers epidermal cell identity. And I'm going to show you also here the expression of this ATML1 homolog, where you see high expression in cluster number 6, and also high expression here in clusters 4 and 8. So, so I took this approach, and what about all of these other clusters? So for this putative nectary cell cluster in banana number 3, it is associated with asparagus cluster number 12. Banana cluster number 1 is associated with clusters 0 and 3 in asparagus. And banana cluster number 4 is associated with asparagus cluster number 2. So let's go back and introduce some of the genes I had shown you earlier, such as the sucrose synthase gene. And here I'm circling in orange the cell clusters that are homologous, as identified from the OMG pipeline. And so what you see here is that sucrose synthase is highly expressed in these clusters. And if you look at the homologous cell clusters here, it is also pretty highly expressed in asparagus cluster number 3, and some expression also in cluster 0 in asparagus. But if we look at other transcription factors, for instance, in this case, this is agamis, you don't really see that pattern. So it does seem to be specifically expressed here for banana. But if you look at asparagus, it's actually expressed in all of the cells and pretty highly too. And that shouldn't be too surprising. If you remember, agamis is a C-class Madstock gene that confers carpel identity. And this isn't just for agamis, it seemed to be the case for many of these other transcription factors that you didn't see, you either saw very broad expression or very little expression, or very little expression, but not in these putative homologous cell clusters. And so as a summary, what I wanted to touch base on is that using this OMG pipeline, it seems to be pretty good at identifying putative homologous nectary cell clusters, for instance, from that example I showed you for the epidermis between banana and asparagus. And what seems to be quite interesting is that these sugar processing genes seem to have shared expression patterns in homologous cell clusters. So they're highly expressed in putative nectary cell clusters, but that doesn't seem to be the case for transcription factors. So I want to end kind of with showing you that, one of the things I kind of mentioned early on, and you might be thinking, like I keep saying putative, or you see these cell clusters, but do I really know if those cell clusters are nectary? And I don't know because I'm losing that spatial information. And there are now more tools being developed to try to incorporate both the spatial context with that single cell context as well. So you get both pieces of information through using spatial transcriptomics. And so those are still kind of hard to do, but it is that next step of being able to get both pieces of information. But even then, with a lot of spatial transcriptomics, you don't get a lot of depth of reads in individual cells. So oftentimes what people do is they do pair these spatial transcriptomics type of approaches with also single cell RNA sequencing, so combining the two. So what hopefully I'll be able to do is, try to confirm, what some of these cell clusters are, either by using spatial transcriptomics, it is just more high throughput, or just on an individual level, just through doing in situ hybridizations or even laser capture microdissections. And lastly, what I really want you to try to take away from all of this, so that was a whole lot of things I just threw at you. But that is just showcasing the power of this type of technology to potentially reveal some information about these different cells and the gene expression profiles. And at least for me, it seems to indicate that the nectary, as many already kind of know, is that it is made up of different cell types. And you have cell types that are important for making sugars, so the nectar components themselves. But you also have other cell types, such as the epidermis or something else that is important then for secreting the nectar. So you kind of see those signatures even in this type of data set. And finally, also that, looking at these conserved nectary structures, because so much time has happened since they've diverged, that there's signatures of conservation and divergence. And really what seems to be conserved are these sugar processing genes. And it shouldn't be too surprising, because if you think about it, the nectary actually is defined by its function of making nectar and nectar components. So I want to just end here, or almost end here, by saying that monocots, it's a great system with trying to understand nectary evolution, really looking at the conservation, looking at then how has it been transferred to other parts of the plant, and then also secondary losses in many of these lineages. And then stepping way back to then the very beginning. So even if you didn't follow anything I said, I hope you come away really appreciating the amount of diversity in nectar and nectary biology, the interactions that it's facilitating between these plants and these organisms, the microbiome, the community in these nectar, the diversity of compounds that can be found in nectar itself, and the structures that that you see in nature. Oh, and if you're interested in learning more, a few people, collaborators and I were putting together a special issue on this particular topic in New Phytologist, and so keep an eye out for this next year. And so I really want to end with then thanking all of the people and also funding source, specifically NSF. And as I had mentioned, it was funded through the collections track. So again, I couldn't have done this without material coming from the Huntington and other researchers at Riverside. And of course, my mentors who I've highlighted earlier, all the people in their labs, even informal mentors such as Felipe Zapata and Philip Sheshkoff, all who have helped me tremendously during my postdoc. So with that, I will end, and I am happy to take any questions if you have any.
Yay, thanks, Irene.
You're welcome. Hold on, let me see how I can, okay, great. I have.
So people are welcome to put their hands up, raise your hand if you have a question, or enter it into the chat. And Peter, I see that you have a question. You have your hand up.
Yeah, so thanks so much. I've never had a chance to think about nectaries in this kind of detail before. And I was wondering, are there any sort of morphological or physiological bases for identifying cell types in nectaries that might correspond to the ones that you've differentiated genetically?
That is a really great question. So I mean, so I've been like revisiting a lot of literature, because actually structurally, septal nectaries have been, examined, characterized quite nicely. And so the more that I look into it, the more it makes me feel like, oh yes, you know, one of the clusters that I'm seeing maybe could have some genes that are highly expressed in the epidermis. I'm like, oh, okay, that kind of makes sense because you need, that epidermal layer, or even some of these other cells that underlie it, how they describe it, they're like, oh, these are parenchyma cells that are, making all these making these sugars. And so there's suggestions from from the literature. But other than that, I think, yeah, I don't know physiologically then how, I mean, actually, sorry, let me step back. Yes, if I can also characterize the nectar, right? So if I were to take the nectar and, run it through like a GCMS or something like that, that looks at then chemical composition, that could also help in, linking the genetics with the nectar that's being made. So absolutely, I basically need more information, right, to then link all of these things up together. I'm not sure if I answered your question completely. I hope so, and if not, I'm happy to clarify that more.
Yes, thanks very much.
Yeah, you're welcome.
Kathleen, you have your hand up.
Yeah, thanks, Irene. I really liked that talk, and I very much appreciated the fruit tart analogy. I think that's the first time I've understood the single cell and the spatial transcriptomics. But I wanted to ask you sort of more of a function question, and like, have you thought about why the location of the nectary moves around so much? Like, I can see why, if you in a columbine that it has to move to the base of the nectar spur on the petal, but in all these other cases, it seems like they're moving the nectary around, even though the nectar ends up at the base of the floral tube. And so why would it move around so much?
That is a great question. That's something I've been thinking about actually with one of my collaborators. Are trying to put together this review, and we're like just trying to figure out how do we put all these puzzles together, right? So as you had mentioned, yes, they seem to be found at the base of flowers, but it seems like that position may be more important, and rather than like, so it may not really matter so much then it's like which part of the flower it's necessarily associated with, right? But as long as it's potentially at the base of the flower where it's kind of hidden, you know, from the pollinators, you don't want it to be too exposed necessarily to pollinators. Um, that is something else to think about too. It's like, well, you know, like you mentioned achalasia, right? So it's at these at the tips of these petal spurs, but it seems like there has to be then some coordination happening between the floral structure themselves to then put the nectary at the tip of the spurs, right? You need the spurs first too to then hold the nectar, right? So it's like what came first? It seems like maybe the spur came first. I don't know. That's something that, you know, something we're trying to think about too. It's like, you know, what actually needs to come first. But the other thing too is like location wise, typically when you look at development of, because you see it everywhere, or in all associated with all of these different flower parts. Typically when you like you're trying to like look at development of the whole flower or something like that, nectaries develop a lot later. And so it seems like it's just like this module that can be taken and put in different places. But how, you know, what is it in each of these species that's telling, you know, that's saying like, oh no, it needs to be at this specific location? We don't really know that yet. Yeah, so big question that no one really understands, knows, and you know, definitely something for maybe, you know, someone to try to understand and look
Thank you.
into.
We have Andy Simpson with his hand up.
I was just wondering, how much evidence do you have that lineages that have that produce nectar lose the ability to produce nectar and then get it back later?
Yeah, great question. I haven't looked into that as much, but there seems to be evidence that suggests you can get that gain of function back if you look at it, you know, at a phylogenetic scale. I don't have really great examples. I think when I was just digging into, like, when I was trying to find examples for something else, it looked like there's a description of not really structural nectaries, but something that seems to be like nectaries, but really isn't, in these Andean melastomates. And the description of it actually was that it was just like this liquid that's, you know, being exuded. There's not really what we would call like a nectary. But if you look anatomically, you find these vascular bundles that are actually found like a few layers beneath. And so that seems to be kind of suggestive that maybe, you know, like there maybe hasn't been enough time to have an actual nectary structure being formed, or there is, who knows, like maybe it just doesn't do that. And you have this more unstructured form of the nectary, and that could be then like a gain of the nectary. So that's something I just recently read. I have to go back and double check if that really is a case of like a gain of nectar function or nectary function, nectaries, but I think there might be a few cases of that for sure.
Cool. Thanks, Irene. We have Justin with a question.
Hey, Irene, I really enjoyed that. And I especially like the drawings. Are those your personal artwork?
Yeah, so I was inspired by other people who drew something similar, and I don't really draw, I like trace. But yeah, this is something that I had put together to try to advertise for our new phytologist call for papers. Yeah.
Nice. Excellent. Well done. Scientifically, sometimes when plants get domesticated, they start to lose like pollinator attraction traits. And I might have missed it. And this is totally a naive, nectary naive and fruity question. But Like, were you using cultivated banana or were you using a wild relative? And is it really super active, or could you be detecting some like genes that were lost? And could you look at things like pineapple and uh, ginger and taro root, like a list of onion and crocus, all sorts of cool like things that are cultivated that may give you like replicated instances of lost then over just a pretty short period of time.
Absolutely. Great question. So let me start off with, let's see, where do I start off with that? So let me start off
Banana.
with the
What kind
first
of
question you
banana?
asked with the banana.
Yeah.
So I'm pretty sure that these bananas, they do produce nectar. So it was, I'm pretty sure, I think it is cultivated. So it actually is. So one of the things I had trouble when I was trying to analyze this data is that it is a hybrid between two species. And so that was a difficult thing, but it does produce nectar. It oftentimes when I went to collect, it was in the morning, but actually for banana flowers, they open and I think they secrete nectar at night because they're attracting bats. Like bats are the ones that come into it. And the next morning when I go, it's like you're just seeing ants all over. They're looking for the nectar there. So it is secreting nectar in that case. And yeah, and I mean, that would be kind of fascinating to see. I mean, I don't know what the major differences would be, but if I were to use something that wasn't cultivated, for instance. But that leads to then you hit up on one of the points. Yes, I am using more cultivated species, and there was a reason for that. One of the things with doing single cell types of analyses, you need a pretty good genome and a pretty good genome annotation. And that's one of the things that I ran into when I was trying to do my analyses. So oftentimes when people are doing this in a completely new species, you also see people reporting on genomes and genome annotations and so forth. So I was relying on what was publicly available, and that's why I targeted these two. In other species, then, like I mean, I would have loved to look at some of these other species you mentioned, like pineapple. So that I did try to do that. I actually have more bulk RNA sequences from that. Not so at the single cell level because I think I was having trouble isolating protoplasts and nuclei. I didn't go into that. That actually took a good chunk of the time trying to figure out what protocol worked the best in isolating these cells. Um, there and yeah, so and I think onion would be fantastic. I did start drawing some up. And unfortunately, I mean, if you think about onion, it has a huge genome, even though it has been sequenced and annotated and everything. So pretty large genome, quite a lot of duplication. So I think that would be also somewhat tricky to try to analyze as well. So yeah, a lot of doing this, it was pretty cool what I was able to find, but it's been really tricky to try to like dig through the data and try to make sense of all of this as well.
Thanks a lot. I mean, the
Yeah,
elephant
you're
in the room
welcome.
are the orchids, of course, right?
Oh my gosh. And I would love to because orchids, I didn't even mention it, but as you had mentioned too, right? Like they don't, I believe they don't really have septal nectaries. And I mean, because there's such a great diversity, you have nectaries that are then found on all different parts, for instance, the spurs themselves. So structurally it's different as well. And that would be really fantastic to try to take a look at. Yeah.
While you've got monkey flowers here, that I don't see other hands up, but if so, I'll stop. There's a report, right, of these erythranthe red hummingbird monkey flowers from the desert Great Basin area that have lost nectar production, and they're reported to like be involved in some kind of mimicry situation where they're growing in sepia canyons with other species in different families that are nectar producing. Having studied columbines, I think of the columbines as like the model, and these things are just like freeloading off the columbines, thinking the hummingbirds thinking that red means they get a reward. And I don't know if that's true, but it's in the Beardsley AFLP phylogeny paper, and he mapped the loss of nectar production. And I know that's outside of your monocots, but you want to weigh in on that?
No, I don't, but I just find that really fascinating too, what you just said. And I mean, I don't know if I looked at that paper per se, but I'll actually be sending, I think my colleague and collaborator should know about it, but if he doesn't, I think that might be something he's interested in, because he's actually, my collaborator, he's actually in, and you might know him actually, he's in Elena Kramer's lab right now. He's a postdoc there, Yan Gang, and he's actually also looking into nectary development and so forth in Mimulus with who was it, with Yao Wu. So I think this might be something that's more up his alley in thinking about the variation that you see. But I mean, that's completely fascinating to take a look at for sure. Well, thank you for sharing.
Okay, we maybe have time for one last question from Kathleen.
If I can just squeak one in, it's a little bit of a tangent, but I was wondering, since it sounds like you've been doing lots of research on nectaries in general, about what's known, like, is there anything known about extrafloral nectaries and how those compare to nectaries in the flower? Are they the same structures? Are they using the same genes, or is it a totally different ballgame?
All of them. So there have been a few studies that look at let's see. So one really nice study has been done in cotton, because cotton actually has floral nectaries, but also then quite a few different extra floral nectaries, so on the bracts and I think on the leaves and so forth. So there's two papers that came out in the past couple of years that have looked at that. And structurally, I think there are trichome nectaries, if I'm not mistaken. I might be completely wrong there. And I think in some of these, it showed that there's some similarities for sure. Like, so some of the transcription factors that are important for nectary development seem to be expressed in both the extrafloral and floral nectary. That is not always the case. So there's also quite a lot of diversity in the extrafloral nectaries, not just in where they're found, but also the shapes. And some of them are more of these like 3D structures. Some of them are more trichome based and so forth. And my personal favorite, I mean, I would love to go and look into this myself, is actually in morning glories. Morning glories also have extrafloral nectaries that are at the base of the I consider them to be extrafloral nectaries. Maybe they're not truly extrafloral because they're at the base of the sepals. So it's on the outer abaxial surface. But in that case, it does seem to function more, what we predict as that defensive role. Like when I saw those in our greenhouse, ants were basically everywhere. And so I'm like, okay, I can see this being the case, out in the forest and so forth. And I think structurally that is definitely different from the actual floral nectary that's in the flower. And so, yeah, no, there's quite a few different systems that people can start looking into to see, is it really the same genes or not? In one case, cotton, it seems like potentially yes, but is that really the case for everything else? So not as many people are looking into it. I know some people have been looking into that, but not as much. I think people are more interested in extrafloral nectaries from an ecological perspective. I've seen more papers there, and that has been fascinating as well, because I just mentioned, this defensive role, but it seems like that might not actually just be the case. There's other hypotheses for why to have extrafloral nectaries. So, yeah.
Cool, thanks a lot.
Yeah, you're welcome.
Well, thanks so much for giving us this wonderful talk and a lot to think about. And thanks for coming, everyone.
Yeah. Thank you so much for your interest and for being here.